Hello everyone. In this module, I will further talk about the bioorganic chemistry part. So, in this section, I will discuss molecular oxygen chemistry, the life with oxygen, and what is the role of oxygen in biology. The dioxogen activation, several reactions taking place with the help of oxygen and using metalloprotein enzyme that is utilizing in our day-to-day life. So, first I will discuss this life with oxygen. So this is the molecular orbital diagram of oxygen. You can show over here. This is the all this bonding anti-bonding molecular orbital. So in the oxygen molecular orbital, if you construct the molecular orbital of the oxygen, you will find that there are two unpaired electrons shown in the red color in the anti-bonding pi star orbital, which are in the same spin, in the same direction. So this is also according to Hund rules, which states that when there are two electrons to be placed in a equal energy, the lowest energy configuration will be that one when the one electron in each orbital will be placed in parallel spin and it will form a triplet state. So that thus the oxygen you can designate can be written over here as shown over here. Oxygen over two oxygen there is one unpaired electron with same spin. So from the molecular orbital diagram, it is very much clear that there are two unpaired electrons residing over pi star antibonding molecular orbital over oxygen. That is why oxygen is paramagnetic. The molecular oxygen that we all inhale, that oxygen is paramagnetic because of presence of two unpaired electrons. So it is a paramagnetic molecule, and it is a triplet molecule because of two unpaired electrons. S value is equal to half plus half because of two unpaired electrons. So spin multiplicity will be 3. So this is a paramagnetic oxygen molecule. Now there are two major important biological reaction that is actually occurring in biology. One is the photosynthesis, where the carbon dioxide, water in presence of sunlight, they are combining and produce this carbohydrate and generate oxygen, which is utilizing the mankind and all the planet kingdom. So this is the photosynthesis part, and the reverse part, using this carbohydrate and in presence of oxygen, the oxygen carrying protein in biology, they produced energy. They take the energy from this carbohydrate molecule, which is stored in the form of carbohydrate. The energy is stored, and they take out this energy with the help of this oxygen carrying protein in presence of oxygen and produce carbon dioxide. And this is called respiration. So there are two opposite reactions that actually happening side by side. One is the photosynthesis and respiration, and both this kind of system are actually carry forwarded by several protein enzyme in biology. I will talk about the most important part, that dioxygen activation, the oxygen reduction to O2 to form this water, proton 4 electron reduction, which is catalyzed by cytochrome C oxidation. Now, if you see the reduction potential, like to reduce oxygen to water, the you require this high amount of energy. This required potential 0.815 volt. So this is a pretty high value. So dioxygen to water, this reduction, 4 photon 4 electron reduction, which is occurring in biology, which is a very high energy demanding process. Still, it is occurring so effortlessly, so easily. So this is the respiration scheme. Reaction is given below. This is a glucose, which is nothing but the carbohydrate in presence of oxygen. This is the oxygen, and it produces this water, and oxygen reduced to produce the water, and glucose is carbon is oxidized to carbon dioxide and produce this energy. So this energy actually is stored in the form of this glucose or carbohydrate, and after this breakdown of this glucose, it produces energy which is utilizing by the plant or animal, all the living kingdom. So now we will understand how this process actually occurs in this oxygen reduction, and which enzyme are responsible for this reduction to takes place. Now before going to analyze the oxygen reduction process, which is catalyzed by metalloprotein, first see that how nature store and release energy. So nature store energy in the form of ATP, which is adenosine triphosphate. So this is a sugar moiety, this is carbohydrate part, and this part is basically the heterocyclic base, and this is a phosphate ester group. So energy actually stored in this phosphate uh, in the phosphate bond, because this phosphoester bond is highly energetic. If you break this phosphoester bond, it will produce energy. So you can see that this is a triphosphate. There is three phosphate which is connected by the oxygen atom, phosphoester bond. If you hydrolyze by water, and it will produce this energy and phosphate ion. You will see that it will produce adenosine diphosphate ADP. From adenosine triphosphate, from three phosphate unit it will produce two phosphate unit and one phosphate ion, and it gives up the energy. So nature actually stored this energy by this kind of molecule. By making this kind of molecule, when it requires, it will break one of the bond and it produce the energy which is sufficient to do some other biological work. So this is a P205 molecule. This is a chemical structure where you can see that there is POP bond phosphorester. This is a POP bond. All this bond is connected by this POP unit. This is a drying agent normally used in the laboratory. In the desiccator we put pure P205, and we can from over the top we can put any chemicals, anything. So basically it is a drying agent. It actually absorbs water. So basically, in P205, this the energy actually stored in this form POP bond. So if you add some water in this P205 molecule, you can see that there is instantaneously there is smoke, or there it will burn out. So it will release high amount of energy. So basically, the storage and release of chemical energy in P205, the energy is stored in the form of this bond energy, in the form of this bond. So when you want to release, you have to break the bond. When you break the bond, if you add water, it will burn this P205 molecule, and it will like giving high amount of energy. It will be formed. So this is the kind of thing, the storage and release of chemical energy. I have given you one example. Now, as I already said that that oxygen to water, this reduction of O2 to H2O, this formation is actually require pretty high amount of energy, because this potential is actually this is actually favorable, because this reduction potential is plus 815 volt. This water to this oxygen to water. Uh, so still, so this is the example of hydroxylation or epoxidation process. You can see over here this all this hydrocarbon, methane, benzene, and this ethene, this kind of molecule. So this is the example of hydroxylation or epoxidation. All this, if you see the enthalpy change, all the enthalpy change are negative, because this kind of change is favorable. This kind of transformation is favorable according to the enthalpy change. However, if this is so much favorable in biology, so then we can see that we human being, or the all the planet kingdom, we are all constructed by this hydrocarbon molecule, hydrocarbon unit. So this, if this oxygenation or this hydroxylation, epoxidation is so much facile, so much favorable, then everything will burn out, because this oxygen can, because the oxygen then will oxidize all the thing, and there will be no existence of humankind. So what is the scenario in that case? What is the main fundamental aspect we have to look into? So one thing is that this potential change, this O2 to H2O, you see that potential is favorable potential, 0.81 volt. Also also this reaction hydroxylation, which is utilizing dioxygen to produce this alcohol or epoxidized product, that is the oxidized product, which is also favorable. So if all these things are favorable in biology, then everything will burn out because of this dioxygen. In atmosphere there is plenty of dioxygen. It will kill all of us, all the humankind, all the plant kind. So then why it is not happening? So let's understand this kind of thing. So the only thing that's standing between you and spontaneous human combustion is a triplet oxygen. Because oxygen is triplet state, because having two unpaired electron, as I have already discussed using the molecular orbital diagram. But are the organic component, organic molecule of all these component part in human being, they are in singlet state. For example, you take that methane or any kind of hydrocarbon moiety that constructed our human body, that are singlet state because there is no unpaired electron, there are no paramagnetism is there. So because of this spin because you can see that because of this molecule, our organic part, which is a singlet, but the molecular oxygen which is available in biology in the atmosphere, which is triplet state. So because of this triplet state of the nature, it cannot react with the singlet state substrate. So because of this simple spin forbidden selection rule, it cannot just react with the singlet state species. That is why the triplet oxygen cannot burn or combust our organic substrate. Although it is thermodynamically favorable, the oxygen, this methane to methanol, all these combustion, all these transformation, epoxidation, hydroxylation is favorable, and oxygen to water, this reduction process is also favorable as I have given the potential energy. But because it is not happening, why? Now because of this spin selection rule, you can understand that this is a, there is two unpaired electron, there is no unpaired electron because this is a paramagnet, this is a diamagnet. So this paramagnet diamagnet species cannot react with each other. That is why stimulated oxygen is safe to us, but the singlet oxygen which is toxic, which is a poison to us. Now how we can generate singlet oxygen, what is the reaction of singlet oxygen, I will discuss in the later slide. Here I have shown the electronic state of molecular oxygen. There are three electronic states. One is the ground state. I have already discussed. This is the ground state where two unpaired electron of oxygen is residing over antibonding pi molecular orbital. This is basically a term symbol that I will not discuss here. Another state possibility is that if the two unpaired if the two electron residing in the same orbital. Here you can see that this orbital is around 22.4 kilocal per mole more energetic, more energy, having more energy than that of ground state. And this is a single state because there is no unpaired electron. All the spin are coupled, all the cancel each other. This is equal to 0 and 2s plus 1 is equal to 1. This single state. And higher than that, there is another possibility is that another electronic state is available for oxygen where two unpaired electron uh, lying in the two same energy level but having opposite spin. Here also you will see that the spin value is 0 and spin multiplicity 1. This is singlet state, and this state is 37.5 kilocalorie more energetic than that of ground state. So this is the electronic state of molecular oxygen where you can see that there is one singlet triplet ground state which is the thermodynamically more stable state is available, and this state, this 22.4 kilocal per mole higher energy in this one delta G form and one this sigma G plus this form is having the singlet state nature. Now if you can generate, if you can give some energy to transform this triplet oxygen to singlet oxygen by giving energy, then it will be very reactive and it will do the all the photochemical reaction that I am coming in the next. Here I will discuss direct spectroscopic detection of singlet oxygen. So this is the organic molecule anthracene based this naphthalene based molecule organic substrate. So in presence of photosensitizer, this is a tetraphenyl porphin molecule. This is the porphin system, TPP system, which acts as a photosensitizer. In presence of oxygen, the molecular oxygen, which is triplet in nature, and light, HNU, the photolight, it will generate singlet oxygen at minus 30 degrees Celsius temperature, 3 hour reaction condition, and CDCl3. So if you do this kind of reaction, you will get ready with this kind of endoperoxide bond formation takes place, photochemical generation of endoperoxide. And because of this endoperoxide is because this is not a stable molecule, it will thermally collapse, decompose, and it will T half is given 5 hour at 25 degrees Celsius. So it will collapse and it will again form the starting material and it will generate, it will form the singlet oxygen. It will form the singlet oxygen. So how you can trap single oxygen, like using this TPP based system photosensitizer, the molecular oxygen in presence of light, you will produce singlet oxygen which will react with this kind of molecule and produce this endoperoxide, and when it collapses it will produce this singlet oxygen. Now when this singlet oxygen, you can see that the singlet oxygen energy level is shown over here, 1 delta. When it comes back to the ground state, triplet state, it will show phosphorescence at 1270 nanometer, this wavelength. So this is a phosphorescence which is occurring because of this singlet oxygen to triplet oxygen formation, and this energy is they are emitting as a phosphorescence, which is shown over here. Now by this way you can detect that singlet oxygen is present in the solution, in the reaction medium. So basically, in presence of photosensitizer oxygen, it will form the endoperoxide which will collapse readily, and it will form triplet, singlet oxygen, and because of the singlet to triplet energy transfer, this conversion, it will show phosphorescent at this 1270 nanometer range. So this is how one can detect that singular oxygen generation process. Now coming to dioxygen activation, we already discussed that 3O2, that 3 means the triplet state, is kinetical inert molecule. The molecule, the oxygen is available around us, is a kinetical inert molecule because it is a triplet state in nature. It cannot do any reaction with the singlet state organic substrate. So it cannot react with biomolecules. So this is the molecular energy diagram I have already discussed earlier. You can see that in the antibonding molecule orbital there is two unpaired electron is shown over here. That is why this is a paramagnetic molecule, or two unpaired electron is there. This is the triplet state, 3O2. This is triplet oxygen. This is the most stable state. So if you can do reaction, it will not take place. But however, if you can put energy and it will cross this energy barrier, and if you can form this single oxygen, for that you require this kind of energy barrier. Delta is equal to 22.5 kilocalorie per mole, and it is for single oxygen. So if you can supply this kind of much energy, it will form single oxygen. Now this single oxygen will do the reaction with this organic substrate, will do this endoperoxide formation, do all this kind of sorts of reaction. So for the dioxygen activation, you have to understand that triplet oxygen which is available in around us, the oxygen we are inhaling, the triplet oxygen which is kinetic inert, which will not react with the organic substrate. That is why you are living. It will not burn us. So but if you can provide energy, sufficient energy, that is 22.5 kilocal per mole, it will convert to single oxygen. Now single oxygen is very reactive, highly toxic in nature. It will oxidize the organic substrate as shown over here. This example, this diene, this alkene, you can say it will form this hydroperoxide, this endoperoxide type bond formation. So for the dioxygen activation, you would need sufficient energy. The energy value is given over here, and it will form single oxygen, and that will do the reaction. Now coming to one of the most dioxygen activation in biology, that is occurring in cytochrome C oxidase. That is the terminal oxidation of this water to oxidation. This is the respiration scheme as shown in the below. Glucose and oxygen converting to carbon dioxide, water I have already discussed in detail. So this is a terminal oxidation, water to oxygen O2 to H2, which is occurring in the cytochrome C oxidase, your terminal member of respiratory chain, having this kind of protein fragment I will discuss in detail in the later slide, having this iron protein and copper protein. Utilizing this kind of metal environment, metal protein enzyme, the oxygen used for proton 4 electron reduction to produce water. So this hydrochrome C oxidase, how the mechanism occur, takes place, what is the active site structure, I will discuss in detail. And in this enzyme, this oxygen activation takes place using this iron copper center. So cytochrome C oxidase that I have already discussed, this is a heme copper. Heme means iron and copper based terminal oxidase, which catalyze the reduction of dioxygen to water using cytochrome C as electron probe. So this is the reaction scheme. Basically, oxygen will be reduced to dioxygen using the proton and electron. The electron will be supplied from cytochrome C. The electron source is shown over here. The sequence from cytochrome C, electron will be flow to copper A site. The structure is given over here. This is a binuclear copper A site connected by this histidine and sulfur containing ligand. And then this copper A site transfer this electron to heme A site. The structure is given over here. This is a porphyrin based system where iron is hexa coordinated with the two histidine moiety. There is no vacant site. From heme A site, the electron will be flow to copper B site, which is the mononuclear copper site, where copper is connected with the three histidine moiety, and this copper B copper B and this heme A3 side. Here actually the reduction takes place, or dioxygen activation takes place. It will bind to this iron copper, and it will take the electron proton, and it will reduce to water. So this is the active site structure. This is heme A3 copper B3 copper B and heme A3, where the actual reduction is taking place. And this copper A and heme A, they are actually supplying the electron in the electron transfer chain. So this is a functional model of cytochrome C oxidase. Active site structure is given. How this enzyme work. From this scheme, we will understand cytochrome C provide the electron to copper A side, which is a binuclear copper side. I have already shown. From the binuclear copper side, it will transfer the electron to heme A side. The heme A is the porphyrin based iron system. To histidine is connected to iron center. This is a low spin heme. This is also transfer this electron to the active site structure. This is the active site structure, the dinuclear center having this copper iron side center. Now here actually this oxygen activation takes place. Oxygen bind to this iron copper, take up the electron and proton, and reduce to water. So this is the chart. This is a graphical chart from where the electron transfer actually taking place, and this the box is shown over here. In this box diagram, this is the active site structure of cytochrome C oxidase. This is a dinuclear iron copper site, but the actual oxygen reduction takes place. The mechanism I will discuss in the next slide. So if you take off the protein ribbon, protein backbone, the enzyme will look like kind of this. This is the heme A3 site, where iron is connected to one of the histidine ligand from the protein chain. Though it is pentacoordinated, one vacant side is left over, where oxygen will bind. And this is a copper B side, where 3 histidine is connected to copper B side, and further it can connect it coordinate to oxygen. So this is the heme A3 structure. Chemical structure is shown over here. This is over this slide. This is oxygen reduction center, actual oxygen reduction center, or this oxygen will take up this electron proton, and it will produce water. Now how the oxygen activation will take place? Now there are several possible mode of oxygen binding. So they are from A to U. You can see that the oxygen can bind to this to metal center by different possible mechanism, different possible way. This is one of the way terminal bridging, eta 1 eta 1. This is actually a new kind of bridging. You can understand this kind of bridging, diamond coat like structure. This is eta 1. This is another kind of bridging binding mode, and there are different kind of binding mode or bridging mode is possible when the dioxygen activation takes place. Now how you can understand this kind of binding using different spectroscopic technique, different stretching frequency. We can discard the other possibility, and we can understand there are like two kind of possibilities there, that I will show on the crystal structure, also evidence from the paper. So actually what kind of binding is there between these two metal center. So although there is different kind of binding possibility, not all kind of binding mode is happening in actual reality. So let's see what is actually really happening in bio. So there are two mechanisms shown over here. First, you can see that this oxygen will connected to this iron 2 reduced form, and it will form this iron 3 superoxide species. Now the iron 3 superoxide species further take up an electron from copper side. It will form copper 2, and this will form this peroxide. So now it will form this iron 3 copper 2, and this oxygen is basically peroxide linkage. Now another possibility is that this oxygen first take up the electron from the copper side. It will form this superoxide this kind of bridging form, and then it will interact with the iron, and it will form this, ultimately give the same kind of iron 3 copper 2 peroxide species. Now there are two kind of possibilities there, mechanism. Now if you look at the actual biology, what kind of structure is there. So this is a very important science paper. You must go through it, and you will understand this kind of what kind of actually binding is actually happening in the cytochrome C oxidase. So this is a crystal structure of heme A3 copper B side of the fully oxidized bovine cytochrome C oxidase. Now if you can look into it carefully, you will understand this iron and copper is connected to this oxygen shown in the red color in this eta 1 fashion. So there are also several possibility. From the crystal structure, it is very much clear that oxygen dioxide is connected to two irons to one iron one copper center via this kind of bridging mode, this terminal bridging mode, end to end. This is called end to end bridging mode, binding mode. So from that understanding, we can propose a mechanism how this mechanism actually occurring takes place, and let's see the mechanism which actually occurring at this heme copper B side in the cytochrome C oxidase enzyme, which catalyze oxygen to water. So this is the catalytic cycle of cytochrome C oxidase. This is the active site structure, the copper B side and heme side. The iron is +2 and copper B is +1 oxygen state in the resting state. Upon reacting with oxygen, it will first form iron 3 superoxide species, which is in turn form iron 3 copper 2 peroxide species, which in turn take the electron and proton, which protonate, and it will form this iron 4 oxo species. This oxygen bond will heterologically cleavage, and it will form this iron 4 oxo species and copper 2 +2 state. And then it will take up the electron and proton. It will form the again iron 3 copper 2 side copper 2 species, which take up the electron, and will form the iron 2 copper 1 species. So this is the catalytic cycle, from where you can see that oxygen will first bind to iron center, then it will bind to copper center, then using the proton electron, this bond will break, oxygen bond will break, the activation will take place, and it will form water. So basically you can say that from oxygen, here you are getting water at this stage. Then this oxidized iron of 4 oxo species will take up the electron and proton, again it will release water, and it will go back to its starting resting state, reducing state, iron to copper 1 state. So basically in this cycle you will understand how this oxygen reduction takes place using these two metal center, and water is liberating, and here proton and electron is supplying by this protein chain. Thank you very much.